In this video, we are going to be learning about biomolecules. And in this year, that is for 2021-2022, you do not have to learn about vitamins, hormones, and enzymes as well. So this is the basic syllabus of the chapter biomolecules. We'll be starting with carbohydrates and moving on to proteins and learning about nucleic acids, that is DNA and RNA. So biomolecules are compounds which are produced by living organisms. Basically, the cells which exist in a living organism produce biomolecules. They can be carbohydrates, they can be proteins, they can be lipids. All of these come under biomolecules. So basically, the reason we're learning about them in chemistry is because they consist of carbon, hydrogen, and oxygen. Most of the time, they consist of organic molecules, right? So it's to do a lot with carbon, and because of that, you learn it in chemistry. So the first one we're going to be learning about is carbohydrates. So carbohydrates, the best example I could think of right now is sugar. So sugar is something we use on a day-to-day basis. It's actually a carbohydrate. Another example could be glucose, glucon D, which we drink immediately to make sure our energies go up. That is also a carbohydrate. So these are basically considered as hydrates of carbon, and most of them have a general formula CxH2OY. This is what they assumed the general formula would be. But there are various examples like acetic acid which followed this, and there were carbohydrates, and acetic acid is not a carbohydrate. Rhamnose is a carbohydrate, but it does not follow this formula. And so the carbohydrate was basically, the definition of carbohydrate was given as optically active polyhydroxyaldehydes. So the word polyhydroxy means that it has many OH groups. Okay, it has some amount of OH groups. It's not one, it's not two, it has more than 3 OH groups. Okay, and or rather, I don't want to quantify it, but it has many OH groups, and it has an aldehyde or a ketone, and they produce such units on hydrolysis. So when a carbohydrate will hydrolyze, it gives rise to polyhydroxyaldehydes or ketones. So as we know, they have a sweet taste, some of them. So sugar is again an example for that. So basically, these carbohydrates, they contain of simple units, okay? And each carbohydrate has, suppose sucrose. Sucrose consists of 2 units, one of fructose, one of glucose. And I'm assuming you have a basic idea as to what monosaccharides, disaccharides, polysaccharides, etc. are. But just to recap, classification of carbohydrates. You have mono, oligo, and poly. Monosaccharides, when they cannot be hydrolyzed to form further polyhydroxyaldehyde or ketone. It can't be broken down more to get a polyhydroxyaldehyde or a ketone. Now, in case of oligosaccharides, they can give 2 to 10 monosaccharide units. So when an oligosaccharide breaks, it leads to the formation of 2 to 10 monosaccharide units. Poly will lead to a large number of monosaccharide units. So they're made up of many monosaccharide units. So there are about 20 monosaccharides which are known, like glucose, fructose. All of these are actually monosaccharides. They're the smallest unit. Okay, but sucrose, on the other hand, is a disaccharide. The reason it's a disaccharide is because it has 2 monosaccharide units. And disaccharides are one of the most common oligosaccharides. Oligosaccharides can also be trisaccharides, tetrasaccharides based on the number of monomer units or monosaccharide units they contain. Polysaccharides, on the other hand, they don't have a sweet taste, and they have many number of monosaccharide units in them. So example of monosaccharide: glucose, fructose, ribose, etc. Then oligo is sucrose, as I said, 1 molecule of glucose and 1 molecule of sucrose on hydrolysis. Whereas you have another example, which is maltose. Maltose, when it gets hydrolyzed, it gives us 2 molecules of glucose. The last, polysaccharides, starch, cellulose, glycogen, gums, etc. All of these are polysaccharides. Now, carbohydrates, as we know, they contain an aldehydic or ketone group. And in the presence of an aldehyde or a ketone, generally they'll be able to reduce certain agents called as Tollens reagent in a Fehling solution. Okay, so if there is a free aldehydic group, they will have a tendency to reduce Tollens reagent and Fehling solution. So reducing sugars, they have the tendency to reduce Tollens and Fehlings, whereas non-reducings do not have the tendency to do the same. Now, but then all the monosaccharides are reducing sugars. This question about non-reducing comes up when we talk about oligo and polysaccharides. So monosaccharides, based on their functional groups, they're classified into two. One are aldoses. Aldoses consist of an aldehydic group, whereas ketoses consist of a ketone group. Now, if they have three, the general term is triose, and if it's an aldose, it's aldotriose. It's a ketose, it's a ketotriose. Next, you have four tetros, aldotetros, ketotetros. You get the idea, right? So these are the basic classification of carbohydrates. Glucose is one of the commonly known monosaccharides, and it occurs as a free molecule, and it also occurs in a combined form. So they're present in fruits and honey, and it's also present in ripe grapes. So if you remember, like glucon D as such consists of glucose. That's because glucose is a very good source of energy. So the various modes of preparation of glucose, let's start with that. First of all, from sucrose. Sucrose is also referred to as cane sugar. So sucrose, when it's boiled with dilute HCl or H2SO4 in an alcoholic solution, it leads to the formation of two things, glucose as well as fructose. The reason is, remember when I told you disaccharides on hydrolysis give rise to their monosaccharides. So sucrose is made up of a molecule of glucose and a molecule of fructose. So when sucrose is boiled, it gives us with dilute HCl or H2SO4 in alcoholic solution, it leads to the formation of equal amounts of glucose and fructose. The second method is preparation from starch. So when starch is hydrolyzed, now starch is actually a polysaccharide, and a polysaccharide is made up of many monosaccharide units. So starch or cellulose, when it's hydrolyzed by boiling it with, again, dilute H2SO4 at a specific temperature under pressure, it forms glucose. It's monosaccharide unit. Next is the structure. So two methods of preparation. One is from sucrose, the other is from starch. Both of them involve the hydrolysis of the disaccharide and a polysaccharide in each case. And in the first case, we're getting glucose and fructose. In the second case, we're just getting glucose. Next is the structure. Now, it's an aldohexose, which means it has an aldehydic group. And it has 6 carbon atoms. That's why a hexose. And it's a monomer of large number of carbohydrates like starch, cellulose, etc. So it is one of the most abundant organic compounds on Earth. So let's start with the structure. First of all, its molecular formula C6H12O6. Now, when glucose is reacting with hydrogen iodide on heating, it is leading to the formation of N-hexane. So the N-hexane formation proves that glucose consists of a straight chain linkage between the carbon atoms. So all these carbons are linked in a straight chain. That is, there is no branching of the carbon chain. The second is the reaction with hydroxylamine and hydrogen cyanide. Now, with hydroxylamine, it's leading to the formation of oxime. And with hydrogen cyanide, it is leading to the formation of cyanohydrin. So here, in this case, you only have the modification at the aldehydic carbon. And this is leading to the formation of oxime. This is forming a cyanohydrin, the aldehydic group. So this is showing that glucose consists of a carbonyl group. Next, oxidation. Now, glucose on reaction with a mild oxidizing agent like bromine and water is giving a gluconic acid. Now, gluconic acid shows, the formation of gluconic acid shows that the carbonyl group is an aldehydic group. Now, think about this. When you learned about the when we learned about the chemical reactions of hydroxide OH groups, alcoholic groups, we learned that an alcohol gets converted to an aldehyde or a ketone, and then it gets converted to an acid. So in that case, we were talking specifically about alcohols, right? But in here, we're talking about an aldehyde group. So this aldehyde group is undergoing a reaction in a mild oxidizing agent. That's why the word mild oxidizing agent is very important. And it's leading to formation of gluconic acid. And this shows that the carbonyl group is an aldehyde group. The next one is acetylation. So acetylation basically means that when glucose is reacting with acetic anhydride, all the OH groups in the compound are getting converted to OCOCH3. They're getting acetylated, and that's forming a glucose pentaacetate. Now this shows, because it's a pentaacetate is formed, this means that there have to be 5 hydroxy groups, and the compound which is formed is stable. So if it had been in the same carbon atom, then we would have gotten a different result. But the fact that the compound was stable showed that the OH groups are present in 5 different carbon atoms. Last one is the oxidation with HNO3. Both glucose and gluconic acid, when they reacted with or rather when they reacted with HNO3 and they underwent oxidation, it led to the formation of saccharic acid. Saccharic acid is basically the conversion of this bottom CH2OH to a COOH. So both the top and the bottom carbon, both of them have been oxidized to give COOH. And this in turn also indicates the presence of primary OH group. So this carbon is bound to 2 other carbon atoms. So this is not a primary carbon atom, but this CH2OH is a primary carbon. Now, the first one we learned about the molecular formula C6H12O6. Second, on heating with hydrogen iodide, we got N-hexane. This meant that it's a straight chain. The 6 carbons are attached in a straight chain. The third showed that we have a carbonyl group because of the formation of an oxime and a cyanohydrin with reactions with hydroxylamine and hydrogen cyanide respectively. And on oxidation with a mild oxidizing agent like bromine in water, it led to the formation of gluconic acid. Now, gluconic acid meant that the top carbon, the CHO, got converted to it underwent oxidation to get an acid. Acetylation in the presence of acetic anhydride, it led to the formation of glucose pentaacetate. The fact that this compound is stable implied that the OH groups are present in different carbon atoms, and because it's a pentaacetate, it also meant that there are 5 OH groups. When it was oxidized with a strong oxidizing agent, both glucose and gluconic acid led to the formation of saccharic acid. Saccharic acid oxidized the primary alcohol saccharic acid didn't oxidize, but the primary alcohol group was oxidized to give us a acid. So this all of the results which were studied, and there were many more reactions which were actually done in detail, and that gave an idea in terms of the spatial arrangement of OH groups. So it was Fisher who gave these these structures, and these are called as Fisher representation. Now, if you see that this is glucose, this is saccharic acid, this is glucose, this is gluconic acid, and this is saccharic acid. Now, glucose is generally called as D plus glucose. Now, D represents the configuration. Okay, but in haloalkenes and haloarenes, you would have learned about things like optical activity. So plus sign indicates that it's a dextrorotatory compound. Now DL, the capital D, capital L, and the small d, small l are actually quite different. Now DL, when we are talking about the capital ones, it's actually with respect to the configuration with respect to another compound. Okay, that's why DL comes up. Whereas DL, small d and small l, means dextrorotatory and levorotatory, and these two are very different from each other. Small d, small l implies that the compound is optically active and it rotates the plane of polarized right to the right or the left based on its based on D or L. So we for carbohydrates use glyceraldehyde. So in glyceraldehyde, this carbon Okay, it has 4 different groups attached to it. There is a CH2OH, an OH, a CHO, and a H. It's an alcohol, aldehyde, hydrogen, and basically a carbon atom. Now, this carbon is going to be optically active because it is a chiral compound, chiral carbon. Because it's a chiral carbon or an asymmetric carbon, it will have something called as optical activity. Now, in this compound, the OH and CHO and H, all these groups will have a specific position in 3-dimensional space. So here you have 2 isomers, plus and minus. So when it's a plus isomer, it's a deconfiguration. Okay, we are considering it as deconfiguration, and the OH group is on the right side. Whereas the minus isomer is the L configuration and the OH is on the left side. Now in glucose, okay, the OH, the topmost OH is on the right side, as well as the bottom OH is on the right side, and that is why it gets a plus configuration. And basically the most oxidized carbon is at the top. So see, this group and this group, they're quite similar. So this means that this gets a D plus glucose as its configuration. Now, the above structures, this structure did not explain certain reactions. First, it did not explain the reaction with Kiss reagent and NaHSO3. Now, it did not form, so an aldehyde group should actually react with Skiff's reagent, but in this case, it did not. Also, it did not form hydrogen sulfide as an addition product with sodium hydrogen sulfide. But so they couldn't understand why that was the case. The second was that the reaction with hydroxylamine. So pentaacetate of glucose, it did not react with hydroxylamine. So this meant that there has to be an absence of this free CHO group. Third point was the fact that crystalline forms of glucose existed. So there's 2 crystalline forms, alpha and beta. So alpha is at 419 Kelvin, the melting point, and beta is at 423 Kelvin. So their modes of preparation were also different. So all of these did not explain the structure of glucose. The open chain was not able to explain these various properties which was observed. And so what they realized was that maybe one of these OH groups underwent a reaction with the aldehyde. So when it reacted with the aldehyde group, one of the OH reacted with the aldehyde, it led to the formation of something called a cyclic hemiacetal. So when the cyclic hemiacetal, is formed when one of the hydroxy group reacted with the aldehyde group. And it was found that the 5th carbon atom reacted, the OH of the 5th carbon reacted with the aldehyde group for us to get a cyclic 6-membered ring. So this ring, actually, this structure was able to explain both the absence of the free CHO group as well as the existence of 2 forms of glucose. Now, if you see, there are 2 forms of glucose, right? There is alpha and beta. Now, both of these were able to be explained by the fact that there is a formation of something called as the cyclic hemiacetal. So here you have the OH group on the right-hand side, and here you have it on the left-hand side. So these are called as basically anomeric forms of, or rather anomers of glucose. Now, the reason we call them anomers is because they differ with respect to 1 carbon alone. So the remaining structure is the same. So 2, 3, 4, 5, and 6, it's basically the same. But the first carbon atom, you have the OH on the right in case of alpha, and you have the OH on the left in case of beta. And this carbon atom is called as the anomeric carbon because only that particular position you have a difference in terms of configuration. Now, the heterocyclic, first of all, heterocycles are the ones which are rings, a carbon ring, which consist of a heteroatom in them. The heteroatom can be a nitrogen, it can be an oxygen, it can be a sulfur too. But there it is replaced. So the carbon chain, suppose benzene, one of the carbon atoms is removed and another atom, a heteroatom, is added. So in case of this heterocycle, this is called as a pyrene ring. So pyrene ring is a 6-membered ring where one of the carbon atoms is replaced by an oxygen. So because the glucose is, is similar to the structure, glucose is also referred to as a pyranose structure. Now here you have the Howard structures. You have alpha D-glucopyranose and beta D-glucopyranose. And you only have a difference in terms of the first carbon atom. And for you to remember, honestly, even when I was in grade 12, I mean, even I finished my master's in organic chemistry, and even now I find it quite confusing to recall how to write these structures. So for ease, I would prefer if you either remember the hydrogen or you remember the OH group. So if you remember the OH group, just remember that these two are on the same side, whereas in this case, it's on the opposite side. So opposite, opposite, and at the top you have CH2OH. That's the only way you can, there's no point in by hearting the structures. It's just that you have to have your own way of remembering it. So with that, we're done with glucose. We started with preparation. We moved on to the structure, and we also understood about the spatial arrangement and the cyclic structure of glucose. Fructose. Fructose is also another monosaccharide like glucose. There's another one called fructose. It's a ketohexose. So the word keto means it has a ketone functional group. And it's a hexose, which means it has 6 carbon atoms. So if you remember, sucrose on hydrolysis gave us equal amounts of glucose and fructose, and that is one of the methods for obtaining fructose. So it is also found in fruits, honey, vegetables, etc., and it is used as a sweetener in its purest form. So its structure, first of all, the molecular formula is same as that of glucose. It's a C6H12O6. Now, based on various reactions, it was found that the second carbon in fructose consisted of a ketone group. So the carbon atom at the first one can't be a ketone because that's an aldehyde. So the second carbon atom is a ketone. Second thing, the 6 carbon atoms are of a straight chain. Again, this is similar to that of glucose, and it's a levorotatory compound. Remember, glucose was actually a dextrorotatory, so you had a plus sign over here, but this is a levorotatory compound, and the D is because of comparison to glyceraldehyde. Now, furan is again a heterocycle which has an oxygen atom, and it is a 5-member ring. So because the cyclic structure, in case of glucose, we had the aldehyde group reacting with the first carbon, reacting with the 5th carbon for us to get a 6-member chain. But in this case, this ketonic group is going to react with the OH group at the 5th position, and this leads to the cyclic structures of fructose. And similar to glucose, you have alpha and beta D-fructofuranose. There it was glucopyranose. Where is it? Yeah, glucopyranose. It's a 6-membered ring, but in case of fructose, it's actually a 5-membered structure. And remember again, the Harvard structures over here, you'll have to learn them. Do not by heart them. Have your own way of remembering these structures. Now, in this case, you will have a difference in terms of the anomeric carbon for us in this case would be, which one? The second carbon. So here, the second carbon atom, you have the CH2OH at the top and OH at the bottom, in this case. In alpha. And in case of beta, we have the CH2OH at the bottom and the OH at the top. Now, the excluded syllabus this time is disaccharides, polysaccharides, and importance of carbohydrates. And so we are not covering that in this recap of the entire chapter. Let's start with proteins. Proteins are the very important bodybuilding foods is what we learn when we are in grade 5 or 6, that we're talking about balanced diet. Now, of course, because it's chemistry, we have to have a bunch of structures in. So we're going to be learning about those structures. So proteins are one of the most abundant biomolecules in the living system. So milk, cheese, pulses, peanuts, fish, meat, etc. All of these are a very good source of protein. Now, proteins actually exist in almost all, or rather in every part of our body, and it is very essential for the growth and maintenance. So proteins, remember, in case of polysaccharides, I said that there'd be something called as monosaccharides, which are joined together to give us polysaccharides. Similarly, in case of proteins, we have something called as amino acids, which will join up to give us proteins. So amino acids they consist of two functional groups, an acid and an amine. That's why its name is amino acid. And they're classified as alpha, beta, gamma, delta based on the position of the functional groups. And here the R implies it's a side chain. So based on the variation of the R group, you have various types of various names of amino acids. Now, when proteins undergo hydrolysis, we have the formation of alpha amino acids. So alpha amino acids are one of the most common one, which means the CH, you have a CH, and both the acid as well as the amine are attached to the same carbon. Excuse me. Excuse me for that. Same carbon atom. So they have a trivial name, which means that it's been, based on the source, or it's also based on based on one of the properties. Like glycine has a sweet taste, and according to Greek language, glycose means sweet. Tyrosine was first obtained from cheese. Cheese are delicious, I know. And tyrose basically means cheese. So the amino acids have both a 3-letter symbol as well as a 1-letter code. So here you have the various amino acids, and you have the R group varying in each case, okay, except for proline. This is the structure of the proline amino acid. Okay, this is the structure of proline, where all of these represent the R group of the amino acid. So, I mean, it's however you can learn it, you need to learn it. And I would suggest, for example, if you could learn it in terms of their classification, like these just contain an alkyl chain, right? These are just consisting of carbon and hydrogen. Whereas here, in this case and this case, you have an amine group coming in. Okay, that is NH2 is here, right? Here you have acids. Here you have an amide. These two are amides. Here you have an alcohol. Here you have SH. Okay, SH is thiols. Sorry. Yeah. Wait, 1 second. Yeah, a thiol. And here you have a sulfur group brand in between thioester. And here you have a benzene ring. Here you have an OH with a benzene ring. So it's up to you as to how you remember these. You have to make up your own mind maps. So amino acids are actually classified based on their nature as acidic, basic, and neutral. Acidic means they have more number of carboxyl group than amino group. Basic, more number of amino group and than carboxyl. And neutral has the same number of carboxyl and the amino group. Based on their essentiality, they are classified as essential and non-essential. Essential amino acids are the ones which cannot be synthesized, and they need to be taken in and through our diet. Whereas non-essential, they need not be, they can be synthesized by the body. So there are various mnemonics to help you remember. For example, isoleucine. First of all, the essential amino acids are isoleucine, leucine, lysine, arginine, methionine, phenylalanine, threonine, tryptophan, histidine, and valine. So these 10 valuable acids have long preserved life in men. So these amino acids, each letter of each word, the first letter represents one of the amino acids. you also have any help in learning these little molecules proves truly valuable. And trust me, this is the only way you can remember. There's no point in learning them in any particular order, but if you remember them like this, it'll be much easier for you to basically be able to study it properly. Next, so the remaining amino acids are oh, arginine spelling is wrong. So the remaining amino acids which are there up here are non-essential. Apart from the ones which are written over here, the remaining are non-essential amino acids. So amino acids are usually colorless, crystalline solids. They're soluble in water and they have high melting point, or rather they are high melting solids, and they behave like salts. Now the reason they behave like salts is because now you have a COOH, you have an NH2. Now NH2 group is considered a base because it has a tendency to accept electrons, and COOH is considered an acid because it has a tendency to lose, oh I'm sorry, tendency to accept a proton, and COOH has a tendency to lose a proton. And because of this, what happens is the COOH group over here will lose its proton to form a CO minus ion. And the NH2 wants to pull away this proton and it forms an NH3 plus. And this is in an aqueous solution. So because of the exchange, it leads to the formation of something unique called a Zwitter ion. Zwitter ion is a neutral ion, so it does not have a charge because it consists of both a positive as well as a negative charge. And this is the reason amino acids show amphoteric behavior. Next is the optical activity. Now, as we know, so this is the structure of amino acids, right? So the R group for all these cases is making sure that this carbon group, this carbon atom is becoming optically active. And this is except for glycine. In case of glycine, this carbon atom has bound to 2 hydrogen atoms. And because of that, it is optically inactive. So all the amino acids except for glycine are optically active. And it's because of the alpha carbon, which is asymmetric. The amino acids can exist as D and L, and naturally occurring amino acids have an L configuration. Next is the structure of proteins. So proteins are basically a lot of amino acids joined together. Before we learn by how that happens, this is due to the formation of something called a peptide linkage. Now, peptide linkage is formed because of there is an amine which is formed on reaction between COOH and NH2 of 2 different or same amino acids. And this is a dipeptide. When you have one amino acid combining with another, same or different, it leads to the formation of an amide bond. Okay. And that is leading to the formation of a dipeptide. So proteins are classified as based on the number of amino acids. So this is the basic idea of the classification of proteins. You have the based on the number of amino acids as dipeptide, tri, tetra, penta, hexa, poly. And based on the molecular shape, you have fibrous proteins and globular. Globular is further classified as primary, secondary, tertiary, quaternary. Secondary is also classified as alpha helix and beta pleats. So based on the number, as it's obvious, based on di will have 2, tri 3, tetra 4, penta 5, and hexa 6, and poly more than 10. Now, polypeptide, which has more than 100 amino acids and has a molecular mass of more than 10,000 atomic mass units, is a protein. Now, polypeptide is less, there are certain cases like insulin where the insulin molecule consists of just 51 amino acids, but its structure is it has a very well-defined conformation, and because of this, it is considered a protein. And insulin is the one which maintains the blood sugar levels in our body. So based on the molecular shape, we have 2 types of proteins, fibrous and globular. Fibrous, as the name, if you think about fibers, you think about like maybe the corn cobs have those fibers which you have, the straight baby corn has those straight lines, and they're basically fibers, or your hair. So they have So fibrous proteins have polypeptide chains that run parallel, and they are held together by hydrogen and disulfide bonds. So these chains are held together by hydrogen and disulfide bonds, and they're insoluble in water. And examples are, I think this is myosin, and myosin, which is present in muscles. I'm sure biology people would know what myosin is. And ketamine, oh sorry, ketanin. Oh sorry, it's actually keratin. Keratin, shampoo, all of these we would have heard about. They're present in hair, wool, silk, etc. Next is globular proteins. So here what happens is the polypeptide is coiling to give a spherical shape. So examples are insulin and albumin. And based on the structure, structure and shape, globular are further classified based on order of complexities. First of all, primary structure of protein. So you have just a straight line. It's a primary structure. So they have only one or more polypeptide chains, and they are linked in a specific sequence. So this is the primary structure of the protein. The polypeptide will have a primary sequence. And if there is a change in the sequence of the amino acids, then you will have a completely different protein which is formed. And that is about primary structure. Next, you have secondary. Here you have long polypeptides that do exist. And due to the folding, you have two types. Okay, you have alpha helix and beta pleats. So alpha helix is a helix. Okay, and to maximize the amount of hydrogen bonds formed, they are twisted into a right angle screw. So there is a hydrogen bond between the CO and the NH, between the oxygen and the hydrogen. And this leads to the formation of a hydrogen bond. Next, you have beta pleats. Now they're stretched to the maximum extension and they're laid one beside the other. And they basically ensure that, again, here also you have hydrogen bonds. And this is to make sure that they basically, the hydrogen bonds are the ones which hold these individual polypeptide chains together. Next is the tertiary structure. Tertiary structure is the overall folding. And you have, so the tertiary structure is from where we get two major, fibrous and globular. And based on the main things that make sure that these structures are stable are the hydrogen bonds and disulfide linkages, van der Waals forces, and electrostatic force of attraction. In case of primary, we didn't even talk about the attractive forces between them. In case of secondary, we said that there's hydrogen bonds, but in tertiary, you have hydrogen bonds, disulfide linkage, van der Waals forces, and electrostatic forces of attraction. The last is quaternary. Quaternary, they have two or more polypeptide chains. First of all, the polypeptide chains are called as subunits. So the subunits will arrange themselves with respect to each other. See, here you have subunits, and they arrange themselves with respect to each other to form a quaternary structure. Okay, so part of that is tertiary, and the tertiary one strand is the secondary, and secondary one line is going to be a primary structure. Last topic under proteins is denaturation. Denaturation in basically proteins are found in the biological systems, and so when there is a change, a physical change or a chemical change, it leads to denaturation. This is because of the disturbance of the hydrogen bonds. As I told you, these three, we talk about hydrogen bonds. And so if there is a disturbance in the hydrogen bonds, it leads to unfolding of the helix, and the proteins will land up losing the biological activity. And so if you remember, generally when we have diabetic patients, the insulin which is present has to be stored in a very cool temperature. This is because if the temperature is very high, it will undergo denaturation. It'll unwind, open up the helix, and then the protein will lose its biological activity. So when denaturation takes place, secondary and tertiary will get destroyed, but the primary structure stays the same because it's just a straight line of amino acids. So examples of denaturation is coagulation of egg whites on boiling. The second one is curdling of milk because of the formation of lactic acid by bacteria in milk. So you have a curdling of milk taking place. And if the milk is spoiled, and that's how you know, when you heat it a bit, you see that it curdles. And this means that the lactic acid is formed by the bacteria in milk. With that, we're done with proteins. So proteins consist of something called as amino acids. You had the classification, we learned about the classification of amino acids, formation of a peptide bond, and classification of proteins as fibrous and globular, and that further was classified based on the complexity. With that, we're done with proteins. Nucleic acids. This is the last concept we'll be learning in biomolecules. Now, nucleus is in a cell. It consists of a nucleus. From what I remember in biology, I learned it only till grade 10. Anyways, the nucleus in the center consists of the genetic material, and this genetic material is responsible to make sure that all this information, this amazing amount of information, gets passed on from one generation to the next. And this, in the nucleus, the reason it's passed down is through a compound, or is through the molecules called the chromosomes. So these chromosomes are made up of two things, proteins and nucleic acids. So there are two main types of nucleic acids, deoxyribonucleic acid and ribonucleic acid. So nucleic acid consists of polymers of nucleotides, and they're called as polynucleotides. We'll learn about what nucleotides are one second. So chemical composition, you have a pentose sugar, a phosphoric acid, and nitrogen containing heterocyclic compound. So this nitrogen containing heterocyclic compound is a base, which we'll be learning about here 1 minute. And the phosphoric acid is again down the screen. The third thing, the first one, that is a pentose sugar, is either a deoxyribose or a ribose. So in case of deoxyribose, it is present in DNA, whereas RNA consists of ribose, and that's why it's called as deoxyribo and ribonucleic acid respectively. Now here, this nitrogen containing heterocyclic is these basically. So DNA consists of 4 base pairs. Okay, before we do all of that, first of all, remember adenine and guanine have 2 rings, one 6-membered ring, one 5-membered ring. Okay, and just remember the basic idea behind these two. Okay, and then remember what the substituents are. So in both these chains, you have this part as a common. But here in this carbon, you have an NH2 for adenine. And in case of here, you have a CO. That is double bonded oxygen. And this CH is the hydrogen is replaced by an NH2. Similarly, don't by heart the cycle. Okay. Remember, all these 3 have the same ring, but they have variation in terms of the things that are attached to it. First of all, the CO is again common for all of these. Okay. And the nitrogen, however, because you have a double bonded oxygen in this case, NH and NH, and this thymine alone has a CH3. So DNA is going to have 4 base pairs, adenine, guanine, thymine, and cytosine. Whereas in case of RNA, it has uracil instead of thymine. So it'll have adenine, guanine, cytosine, and uracil. The structure of nucleic acids is very important. So first of all, you have something called as a nucleoside. Nucleoside consists of the attachment of the base to the first position. So at this first position, you have the base attached to it. And the reason we are numbering it as 1-, 2-, 3-, and so on is because it makes it easier for us to number the carbon atoms of the ring. So here you have this is a nucleoside because it just has a base, but a nucleotide has a phosphoric acid. So a phosphoric acid is attached at the 5th carbon atom. So instead of a hydrogen, you have a phosphoric acid group attached to it. And nucleotides are joined by the phosphodiester linkage between the fifth position of one pentose and the third. Okay, not to complicate this. So at the first position, we know you have a base attached to it. So this is the first, second, and third, fourth, fifth, right? So what happens is there is a bond formed between the fifth carbon, the phosphoric acid, this part, and this, the third carbon. And this leads to the formation of something called as phosphodiester linkage. So the third and the fifth carbon at the 5' and the 3' position of the two different sugars or two pentose sugars is leading to the formation of a nucleotide, a dinucleotide. Okay, this is the basic understanding. So this sugar is bound to a base. It is forming a bond with the phosphate, which is forming a bond with the other sugar, which is attached to a base and a phosphate group. So in case of primary structures, you have the information regarding the sequence of the nucleotide. Now, in case of a secondary, so you have the, the nucleic acids, the nucleotides can exist as a secondary structure as well. And that is where we have this beautiful double helix. So a double helix structure was actually given by James Watson and Francis Crick. So what they said was that the two chains of nucleic acids are wound together and the bases are going to form hydrogen bonds in between them. So remember, in case of our secondary and tertiary structures also, we studied about the presence of hydrogen bonds between the various amino acids. Similarly, in this case as well, you have hydrogen bonds between the base pairs. So guanine forms a triple bond with cytosine, adenine with thymine. And no matter what they form, because these are going to be complementary to each other, these chains will actually, the base pairs will always be this. You will not have guanine binding with thymine. It will always be guanine and cytosine, adenine and thymine in case of DNA. In case of RNA just will have one strand. It does not have two strands like DNA. It only has one strand. And it folds back on itself, and it based on its function, RNA is of three types: messenger RNA, ribosomal RNA, and transfer RNA. Now, this is very important, the biological functions. It is responsible for heredity, and it reserves the genetic information. It is also responsible for maintaining identity of various species and organisms over millions of years. And it is capable of self-duplication. Identical DNA strands are transferred to the daughter cells. And nucleic acids are actually responsible for the synthesis of proteins. So only based on nucleic acids, you have the ability for synthesis of proteins. Because as I told you, the amino acids, if the order of the amino acid varies, then there will be a different protein altogether. And I think on some level, learning about all of these is like one natural, it's mind-blowingly beautiful. And there are various methods by which they make sure that the amino acids are arranged properly, and they check that as well in the cell. And if that's not magnificent, and I know what is. The proteins are synthesized by various RNA molecules, and they help in the synthesis of a particular protein which is present in DNA. Now, DNA fingerprinting, as we know, fingerprints are unique for every individual. So unless and until you can surgically remove it, Men in Black style, you can, that was basically burnt his fingertips. So that will make sure that the fingerprints are removed. But DNA cannot be removed. Every single cell in your body will have the exact DNA, and that cannot be changed. DNA is actually provides a multitude of information regarding a human being. So uses of DNA fingerprinting include forensic labs to identify criminals, determine the paternity, identify dead bodies and accidents, comparing them to the parents or children, identify racial groups to rewrite biological evolution as well. So DNA actually can, if we are able to progress in the right direction, DNA can actually help identify what kind of medicine can be given to one person, and that might be good for one person, might not work as effectively for another one. So that, and DNA also provides information if a person is susceptible to various diseases like cancer, and DNA is like one information box of that particular person. So there are two scientists we need to talk about, James Dewey Watson, Hargobind Khurana. About those respective scientists, it's given here and you can go through that. With that, we're done with uh, biomolecules. We started with carbohydrates, moved on to proteins, and learned about nucleic acids. This year, that is for 2021, 2022, you do not have vitamins, hormones, all of that in your syllabus. So with that, we're done with biomolecules. There are other videos where you can check out the other chapters for this term. So if you're, and also one more thing, you have the notes in the description below.